Zoom recording. Okay, well, welcome to those in the room. I'm sure we'll have a few that trickle in as we go along. This is Oregon BHA's webinar or virtual Pint Night with Oregon Department of Fish and Wildlife on CWD. Most of our membership is very aware that Idaho detected several animals, both deer and elk, with CWD, and so it became a larger concern, or a more immediate concern, I should say, for the state of Oregon, considering that's not far from us. So the ever slow march of CWD goes on. And so we were really fortunate to get to connect with Colin and Dawn of Odfw to really give us a presentation. Colin, you're the veterinarian, is that right? The statewide?
I am state wildlife veterinarian for Oregon.
Right. That's right. And then Dawn is the Unglate Coordinator, if I'm saying that title right, hopefully, or close to it. All right, perfect. Dawn, you do have yourself on mute at the moment, just as an FYI.
It's safer that way since you're recording.
Trust me, that's, like I said, once we get started, really. So really fortunate to have these guys and just a crazy amount of knowledge, obviously, and experience in this. And of course, working with the public in, the prevention and management of CWD, and to go over Odfw's plans and how we, as Oregon BHA and our membership, and even outside of that, can really help support Odfw in both the prevention and management of this critical prion. Again, and I should add real quick, I'm Joel Strimling. I'm the communications coordinator for Oregon BHA. And, if you ever have questions, feel free to reach out to any of us at BHA. But otherwise, I am going to hand the microphone virtually, so to speak, to Dawn and Colin to give their presentation. And then after that, we will certainly have some questions and answers as well. So I'm going to mute myself, gentlemen, and let you take the floor.
Great. Okay. Well, thanks for having us. We're going to give you an overview of CWD quickly, and then go into a breakdown of our Oregon CWD plan. And then we'll just finish up with some sampling. Don's going to go through some sampling of how we foresee us moving forward and why we need to sample, which is you'll be pretty impressed with the maps that he's put together and the data. And then we just kind of open it up, what you can do, what we can do, where we go from here type of thing. And that's at least the plan here. This is, you may have, some people may have seen parts of this. I put together this slideshow with several people, and it was huge. And we gave several of these presentations internally to researchers and managers, and talked for an hour and a half or more. And so we've cut this down quite a bit. And having a group like this to talk to is great, because it helps us fine tune the message. You can get super technical, as you well know, with a topic like this, and it's good to keep things on the level, and so everybody understands it, including us, and just so we can point towards what our mission is. So I will I'll start my screen share here. And there we go. So this is where we'll start, and really this is kind of the title of our plan, the Odfw plan, and it's literally for prevention, surveillance, management, and control of chronic wasting disease. It's our first plan was put together by Don Whitaker back in 2002. We actually started sampling a handful of deer in 1996. We started putting together plans, as did the entire nation, right around the beginning of 2002. If you had CWD or you were worried about it, you started putting a plan together. We had updates to this in 2005, and tried to do it every 5 years. These have all been internal plans. They weren't something that we ran through the commission or the legislature or something like that. These were internal plans. We were the furthest west, and thinking maybe one of the last to get CWD. So we looked at all the other states, and the Oregon plan is an amalgamation of many plans actually, and our own. It has, of course, a lot of unique stuff in it, but we had the benefit of Colorado and Wyoming, and even our Midwestern state partner states, as well as some of the federal and other plans for state properties and the like. So this plan is we've been putting together for the last, at least this version for the last 2 years, with a small group of individuals and kind of a working group, folks around the state, as well as we had a member of the Oregon Public Health Authority on board, and we met several times prior to COVID hitting, and then actually put this thing together, and it's gone around the state several times, and it's not a secret plan. It's public facing, but it's not something, again, that we have that we have set up as an official document that has to be ratified by the agency. It's a guidance document is what it is, for us to be able to move forward and do a variety of things if we need to. So this is the latest map. Well, there may even be another one, but this is the latest one of that Brian Richards with National Wildlife Health Center at USGS there in Madison has put together. This has been the map that everybody uses. It's a really good one. We're looking at this now with that gray area up against our northeast corner. And, we'd been expecting it for a while. It just was kind of surprising. I think it's probably surprised everybody on this call that it just arrived. It's a type of disease that is very hard to find in the first place. It takes a lot of effort. It generally occurs at a low enough prevalence that you can look in lots of areas and with lots of, even if you have it, and you may not find it. So we've been sampling for quite a while, and we'll go over some of that. So it's a transmissible disease. It's called a spongiform encephalopathy. It creates holes in neural tissue in the brain that you can see on a microscopic slide. And it is primarily a disease of deer, elk, and moose of North America. There are a handful of other species that are cervids. It's a disease of cervidae right now. And it's characterized specifically by animals wasting away. And for all intents and purposes, it's 100% fatal. So it's picked up almost 100% orally is the way that cervids pick it up. I mean, it can be inhaled, and there could be other uptake. There's some thought that there could even be cases of transmission to fetuses, but for the most part, the disease is picked up orally through the prion being either on an animal or on the ground or on a food item or in a contaminated surface. It makes it through the gut. It's picked up in the intestine, and it works its way at that level into the neural system from the intestinal bloodstream, and eventually ends up in the brain. It's a lot more complex than that, but that's basically oral room, it goes through the 5, including the gut, the multiple stomachs and the intestine. So the organs that are affected by chronic waste disease are primarily the brain. And we'll have a couple slides on this. The prions can accumulate in the eyes also. And you could actually test for it in vitreous humor of the eye. We don't, but you could. And you can also pick it up in high concentrations in the spinal cord. And one of the things we test for, which we'll talk a little bit about, is lymph nodes, particularly the lymph nodes that drain the brain, the portions of the brain that we look in for. Those are the 2 main areas that we get the initial, at least detection when we're running diagnostic tests for the prion. So this area, this will get more and more detailed, but it attacks the brain. And one of the areas that it sets up shop is along the spinal cord, just underneath the cerebellum. And within this area, this brainstem area, is where we test for. So in this kind of cartoon schematic that came out of the Denver Post many years ago, it kind of gives you a view of what you generally would see under a microscope, that when you're looking in an understained section of brain, if you see a lot of holes in the tissue, it means the cells have died. And so that's one of the things that they see. They see staining and they see holes within the sliced sample that's sent to the pathologist. So similar diseases, CJD, Creutzfeldt-Jakob's disease. There's a sporadic form that occurs in humans, and it occurs in all states, and at some percentage that is relatively microscopic, but it still occurs. There's a variant CJD that comes from mad cow disease. We've all heard of that. And it's actually called the new variant CJD. And then, of course, Spongiform Encephalopathy, and then sheep and goats. And there's other ones besides this. There's other species that have spongiform. Probably all species have a spongiform encephalopathy, either that is current or they've evolved to be able to withstand infection by it. The scrapie one, this kind of entries, this is a scrapie sheep. And so if folks ask, why do they call it scrapie? And one of the manifestations of this prion disease in the sheep and goat version is that it makes the skin, extremely irritated and itchy. And the sheep will just rub their entire hide off as part of this, and be neurologic and everything else that the diseases do. So, quick history, 1967, possibly even before that, if you talk to Mike Miller out of Colorado on this, is where this first started showing up in Colorado. They have a research facility there. The research facility, prior to being given to the Colorado Department of Wildlife, was owned by it was run by the Department of Agriculture that did scrapie research. So CWD looks a lot like, at least from an electron microscopic standpoint, looks a lot like scrapie. And one of the theories is that it evolved from scrapie, maybe at the Colorado Research Facility, maybe not. It could have just been on the landscape. Wyoming and Colorado have a lot of sheep in that part of the country too. So, so the other site that we saw this was in Wyoming. Started seeing affected animals in their pens a couple, about 10, 12 years later at the Seville Research Center out of Wheatland. And then it was first identified actually in free-ranging elk in 1981 in those areas, northeastern Colorado, southeastern Wyoming. Seen in free-ranging mule deer. Then we started. There was a case that the states would actually send research animals around to other states and provinces to be used in research or to be used in elk farming, which was not a good thing, of course. And this is before they even knew what this was, or that there was a prion disease even had a name. So part of the spread was by the agencies early on. During the late 90s, of course, we had you can see a kind of an explosion of farmed animals that started showing up with this. And then lo and behold, we sent it to Korea, to South Korea in 1997. They have it they still have it in several of the areas where they raise cyca deer. So since then it's grown to multiple states, and we just looked at that map. It's in farmed cervids, it's in actively farmed cervids, as well as it's spreading and on our doorstep. There's really no evidence of CWD showing up in non-cervid species. And the known are of course, the North American cervids, as well as reindeer, caribou, likely, as well as cyca and red deer. What's interesting is this, even our wild species, mule deer, black-tailed deer, multiple subspecies of whitetails. Of course, we have multiple subspecies of elk. They have different susceptibilities and vulnerabilities genetically, and that's one of the research things that's going on now to try to see. One of the things we do know is white-tailed deer are tremendously susceptible and vulnerable. We think about mule deer having it because of Colorado, Wyoming, but they're actually less susceptible. If you put them in all things equal in a pen, they're going to get the disease a lot less frequently at a lower infection rate within a pen. So, but nonetheless, if you leave them in there long enough, they're all going to get it. So it's transmissions nose to nose. I mentioned it. There's some research so they could be passed in utero. Environmental contamination is a huge issue and challenge. Probably the biggest challenge in all of this is the fact that it can lie for decades. We actually don't know, but we don't have any examples of land that's been highly contaminated that is not now highly contaminated. So it's just something, I mean, nothing could be worse. If you have some things that you don't want with the disease, one of them is that it contaminates the environment such that an animal walks across it and gets the disease. It's one of the worst things you can have. Transmission nose to nose, there's lots of diseases that do that, they come and go. And this environmental contamination has kept this on the landscape and helped it spread. Its incubation period is long. It's over a year for mule deer, about a year for elk on the average, and up to 2 years in mule deer. And for elk, there was an elk that was in the Wyoming research facility that finally died without it testing. It was in the most contaminated pen its entire life, and it never showed signs of CWD and did not test positive. So infective animals is shed via saliva, feces, urine. It's in their blood, melting carcasses, and infected tissues are also just as infective as saliva. The time from infection to shedding is unknown, but it could be fairly quick when animals are asymptomatic. That's another bad. It's like with some of these viruses that we get. You hear about all the time. Well, you get a certain virus, a cold virus or influenza or something that you're shedding for a week before you show the first signs. So that's another thing that you don't want in a disease. Well, this happens in CWD. Take home on this, just it was 100% fatal. Mentioned that there's no treatment, there's no vaccine. There is research presently for the first time that Wyoming's doing, looking at a vaccine. I don't know anything more about it. I know it's supposed to be at one of the conferences here this summer, an update. There's no cure. It is the most, of all those prion diseases that I mentioned, this is the most infectious. It is the worst one of all of them. It's the most transmissible, and it has the most bad qualities. Some of the prion diseases are not, do not have an environmental component, or it's a very weak environmental component. Some of them are just hard to pass. The human form of CJD, about the only way you can get it is if you consume a person's brain, which there's a form called Kuru that happened in Papua New Guinea years ago, where consuming human brain was considered part of their culture, and it had a pretty high percentage of CJD in the population before this was figured out. So the human health side, that's the only thing I'm going to really say about this. There's been experimentally infected squirrel monkeys, lots of research on mice that have been modified to have the human genes. There's macaque studies that have been done multiple times that have shown, some have shown that transmission is possible. One study that did this out of Canada fed the macaques. And these are crab-eating macaques. It's like a subspecies of macaque that has a very high correlation to the human genome. And if you fed, it would be the equivalent of feeding a person like a 7-ounce highly infected steak, once a week or twice a week for a year. And that was the level that they were consuming this. And apparently several of the macaques actually had prion forming in the brainstem area. They tried to repeat this study, and USDA, the research group out of Ames, Iowa, tried it, and they couldn't repeat it. They couldn't get it to work. So who knows? The latest research out there related to humans is they did a lot of studies with these mice, these humanized mice, and they would inject CWD in their brains. They would give them all kinds of CWD in various ways. So the mice were a surrogate of us because they had a lot of our genes, and it kept showing that they had no uptake. Well, the most recent study is they actually they were killing the mice. The original studies, they were killing mice, say, at a year or at 9 months or something. Well, they doubled that or tripled that time. And lo and behold, they started finding, if you let it go long enough, the mice actually got CWD. So, but what's that mean to people? There's no evidence the occurrence of CWD in humans from consuming cervid meat, and there's been lots of people looking into this, including the prion National Prion Surveillance Center in Cleveland at Case Western. They get every CWD or every CJD, the human form, sample that they get their hands on. They do a history on if they can get it from the family. Did the person hunt? Where did they hunt? Did they were they successful? You know, did they consume deer and elk meat, that kind of thing. And then they look closely at the human form to see how closely it matches the CWD form. And so far, they've seen no uptake by humans. So that's good to know. Honestly, this is the last public health slide. We tell everybody to wear protective gloves. This is from the CDC. Wash your hands, disinfect field equipment. There's lots of stuff out there on how to disinfect with bleach on field equipment, knives, and various items we use in dressing out our animals. Avoid sawing through the brain or neural tissue. Try not to cut through much bone if you can, but just be mindful that if you could possibly have a CWD infected animal, you want to try not to spread the prion through infected tissues. And again, don't, this is obvious, don't consume meat from animals that appear sick ever, or are found dead of unknown causes, particularly in a known CWD infected animals or in areas that are infected. There are 3 transmission ways that CWD spreads. There's natural, of course. They can walk across the border, and we may get our first case that way. It's not very far from our border, just through mule deer migrations. Hunters, of course, can move infected carcasses. This has happened, at least believed to have happened, several states. And then the fastest way it gets spread is in a trailer. Move infected live animals. We do not move ungulates. We haven't for years now. And I'd say that I'll qualify that. If we move something, it's within its home range. We don't move anything out of its home range. So nothing from the north coast to the south coast, you know, that kind of thing. That we're real careful with where we're moving things. So it needs to be within a herd unit or a herd unit or an animal's home range, at least whatever the biologist believes that to be. So how prevalent? I think it's 30 states now. Louisiana just came down with it. There's several new states, 4 Canadian provinces. Higher in free-ranging deer than elk. So again, it's the species difference. Adult male deer always are more infected, but it's likely related to behaviors of male deer versus female deer. Vehicle-struck animals. If you have an infected area, and you know there's CWD animals on the landscape, and you're going to end up with more animals being hit that have CWD, and they're just not right neurologically, so they can't get out of the way quick enough, so they get hit more often. How close? We talked a bit about this. Less than 25 miles. Idaho tested 6 positive animals out of approximately 530 harvested. I think they got a few, maybe 100 more than that. But anyway, they have it in 6. The bad news is they found out of 6 animals, they found it in 3 species, which is really bad news. It's probably more widespread. It's probably it takes. And I don't have any information on this population for this, but in general, biologically, it takes years for it to spread between species, and or it takes years for it to be on the landscape that all these animals, these different species, would be using the same highly infected pasture in order to get this. So it's been in western Idaho on our northeastern border for a lot longer than last fall. So, and the other bad thing about it is it likely is, it makes it more likely to be in Oregon right now. So that doesn't make you sleep any better. So anyway, surveillance wise, we target surveillance looking, our targeted surveillance is looking for sick animals. The animals that just look sick, they're progressive weight loss, they're skinny, their ears are droopy. And we see this every month or week, and we take them. Yeah, we take them, we test them, and it's something else. It's been that way for the 25 years that I've been doing the vet thing, and it's just it's good to sample anytime you see a sick animal. It doesn't, particularly with CWD now. And if you see something, call us, and we do want to test it. That's for sure. Other signs, teeth grinding. You can actually hear this. We had a calf elk not long ago that was in a small. It was leaning up against somebody's truck, was neurologic and grinding its teeth, and you could hear it on the phone video. It had excessive salivation, and it didn't have CWD. It actually had pneumonia, but it had a variety of things wrong with it. So clinically, symptomatic animals just look sick. And all the pictures that you and I have seen are the same pictures we see on the internet from Beth, a lot of them from Beth Williams and Colorado and Wyoming. You know, they're lowered ears, they're blank expression. It's a neurologic look. They're trying to die. You know, the respiratory distress thing is they've inhaled ruminal fluid and they have infection in their lung, pneumonia. So what do we test? Particularly with elk, but we can do this in anything. The gold standard test has been looking at the obex. And the obex, I don't know if you can see my pointer, this middle bottom, the obex lies right under the cerebellum in the brain. This is the back of the skull. This is the front. And if you remove the cerebellum, this lies about an inch inside the foramen magnum hole. You know, inside, you pull off the first vertebrae, you got the back of the skull, lies about an inch in there. And we go in there with a grapefruit knife and reach forward and cut this off on the front end. There's side nerves, the 9th, 10th, 12th cranial nerves are right off the side here. We cut those off. They're just little nerves that come off the sides, hit into the bone. This is what we submit to the testing labs. Okay. The other thing that we test, which we test even more, is the medial retropharyngeal lymph nodes. And you can see where the antler is here. This is going in at the right at the throat, the Adam's apple level of this male deer. And the medial retropharyngeal lymph node lies right on top of the trachea. Okay. And so what it drains is that obex. So if you're going to sample two things, you want them connected. They're the ones, those are two tissues that get the prion the earliest. When animals are asymptomatic, you can find CWD prion through staining and other testing. So those are the two. I took some things out. We test all kinds of tissues too, that if you can't get those two, we can get tonsils. We can look at lymph nodes of the gut, the mesenteric lymph nodes. You can take the rectal tissue right around the rectum, has a bunch of lymph nodes. Those will also show lots of prion. But we almost never pull those tissues. It's almost 99% the medial to retropharyngeal lymph nodes, which are not all that difficult to get, and or the obex. Okay, So Oregon's plan is based on Association of Fish and Wildlife Agencies best management practices. This was written in 2018. I was the lead editor on this. It had 30 veterinarians and biologists from around the country. All the ones you'd want on a best management practices, and it's in there's a supplement to it that says 2019, and these two items are if you Google best management practices CWD, it'll take you to this if you haven't already seen it. And so it's a great document. It's nothing more. It's not prescriptive. It's guidance again for the states of what works best. And one of the reasons we did that was because the states, some states, this didn't include Oregon, but some states would get a lot of political pressure from their legislators or others to not do certain best practices as far as testing, or you could be a variety of things really. And we put this together so that the directors would have something to take to the legislature and say, well, you can make that decision. You're the legislature, you're the governor, but here's the best practices it's approved by all 50 state directors. So it's probably one of the most powerful and handy pieces of work that can be used by the leads of all our wildlife agencies, and they've all applauded this thing. It's great. We've had about 50 authors of the various chapters. Okay, so here's our mission. Of course, this is pretty obvious. We want to prevent CWD from affecting Oregon wildlife, and if we have it, we don't want it to spread. We don't want it infecting new herds. So if you keep that in mind, that's our goals. We want to keep it out of the state, but when it does come in, walks across in a mule deer, we want to try to do everything we can to keep it from spreading. Not that every state hasn't tried this, and there's a variety of ways that every state has approached it. They have different rules, different laws, different populations. I mean, it depends on where you get your first animal. Michigan got their first CWD deer in a neighborhood in East Lansing. And what are you going to do with that? Of course, they removed every deer they could in the neighborhoods, but then they started seeing it in other places that radiated out from there. Because by the time you see it in a neighborhood, in a symptomatic animal, it's been on the landscape for 2, 3, 4, 5 years. So, okay. Our 4-pronged sampling strategy is targeted surveillance, live sick-looking animals. We hope we don't test. That's the worst way to find CWD, because again, if you see a sick animal and it's CWD positive, it's been sick maybe 2 or 3 years and dropping CWD in feces, infecting the landscape, and it probably got it from something else. So it's the worst way to actually find it, but we'll take them all. We got to get them off the landscape. Hunter harvest animals or through damage removals, we're testing all those. Our best sample is from hunters. By and large, by far, hunter harvest is the best samples. We get the most that way. I'll talk a little bit about roadkill salvage, but we're getting, you have to turn in all the roadkill salvage heads, and we try to sample all the adults that we get. And then any cervid that dies in a elk farm, of the 8 elk farms we have, we test all of them. So targeted surveillance should not be the primary surveillance method. If you see one, it's been on the landscape for a while. So it's clinical signs. Here's our hunter harvest. We use weighted sampling. We're looking at male deer. A male deer that you test is worth 1.5 female deer, and roadkill, because roadkill deer have a higher chance of being CWD positive in a CWD positive area. They're like worth 2 points more than just any animal. Our state borders we sample for sick animals. We're now focusing on Northeast Oregon, and that's going to be our sample focus. We're going to try to test. We haven't actually increased our harvest. Like we're not trying to nuke the populations Northeast Oregon or anything, but we're really going out of the way to try to get more samples up there. I mean, if we could sample every animal that's harvested from Northeast Oregon, it would be awesome. But Don's going to talk about that in depth. And we're going to continue random sampling from any sample we get anywhere in the state. Because when you think you know where it is, you got to figure it out. It's like anything. It shows up somewhere else. So we want to just do the best job we can. And we're looking at all adult cervids, does and cows, cow elk. We don't hunt moose, but we're testing any moose that gets hit on the highway or dies of some reason or another. And with deer, it's really our best chance of finding CWD is with an animal that's at least 2 years old, of course. But a certain percentage, maybe if you had 100 infected yearling deer, you would find it in probably 20-25% of them. So they're still valuable. So we sample as many deer now as possible, at least a year of age. Male deer is a premium, all adult elk, cows, as well as bulls. Roadkill sampling. This is statute enacted by the legislature. We said we had to do this, and then we were able to add administrative rule to it, which is really a benefit, particularly for us hunting and hunters. And so what we did was we added to that. Okay, well, this you could collect roadkill animals. Here's some guidelines. However, you have to number one, get a free permit, which you do online. You can do on your phone. And you have to turn the head in within 5 days, and then we can test it. So this is a tremendous sampling tool for us statewide, because deer, they get hit all over the place. They get hit more often during migration, seasonally, but they still all year long, these things are getting hit. So, and from one corner of the state to the other. So here's people say, well, how much have you sampled? We've sampled over 24,000, and we've added another 400 or so to that this year. But it's we've collected usually between 800 and 1,500 animals, which is pretty good. We've put a lot of effort into it. Some areas we do a lot better than others. We do really well in Western Oregon, and it's just harder to collect in Eastern Oregon. We're going to get better at it. We're going to figure out ways to set up more check stations and try to work with groups like Oregon backcountry hunters, and try to come up with some good ideas on how to sample the eastern side of the state a lot better. And then farm cervids. Oregon is a leader in sampling farm cervids. We don't have very many, so it's easy to be really good at it. But we sample all suspicious deaths. Do a complete necropsy, and any animal, doesn't matter if it died from an accident or it went to slaughter. We sample everything over 6 months of age that dies of any cause, and we're the only state that does this. So effective surveillance requires statistically significant sampling. The inset picture of all these vials of CWD, that is in Oregon. That's a shipment that's about to go out that our lab coordinator, Richard, was about to ship out, and we took a quick picture of it. Just to give you an idea of how many, we do multiple of these in the year. The bigger picture is, I think, is Michigan. And they sample up to 40,000 heads a year, which is amazing. They have about 20 staff that do nothing but sample heads. So the other thing that adds to being very effective is and important to me as a hunter, is we're putting together an online CWD results reporting site so that when you harvest your animal and we take a CWD sample, we'll give you like a card with a phone number and we'll track it with your hunter number, and then you can call in or you can go online, and there's a variety of ways to do it. You can do it with a QR code with your phone. You can do all kinds of things, and so within a week to 3 weeks, 4 weeks, depending on how many you get in the middle of the season, it slows down getting results, but you'll be able to get your results real time, same time we get it. So that'll be great. And we'll have that up and running by this hunting season. So prevention is our big thing now. And, I'll give you some examples, but it's things such as outreach, talk about regulatory tools, animal density planning, scents and lures are tied in with some of these regulatory tools, carcass disposal. We'll talk a little bit about some of these quickly. Outreach, we're doing this tonight. This is good. This is great, actually. So in this week, we need to do more of these. We've had 2 or 3 talks with our constituent groups, and that's awesome. But we're working on updating the CWD web page. We've updated it several times in the last couple of months, and we're going to continue to do that. We're going to do some blog stuff with Tim Akamoff, who does the CWD or who does the ODFW blog page, and we'll see some more messaging in the regulations. We already have a page in there every year, but we're going to beef that up a bit. So regulatory tools, you all, I'm sure, are aware of this. Here's the roadkill salvage. It went in effect in January of 19. Mandatory check stations way back early on. It's been voluntary for I don't know how long, 30 years or more, and now in January 2022, we finally were able to pass this for the sake of CWD, for testing CWD. The legislature said, yes, you can have a mandatory, if you got a deer or an elk in your truck and you see a check station sign, it's mandatory to stop to have it tested. So it's great. It's good for sampling. It's good for Oregon. The legislature decided they did not want urine attractants back in 2020. This was not something that we took to them or anything. They just wanted, the subcommittees decided they wanted to do that, in a way they went with it. We had the parts ban as far back in 2002. Don set that up and passed it. We've had cervid sampling, fencing since 2009 with mandatory sampling. Wildlife rehab cervids has been heavily regulated. In fact, we have no, for all intents and purposes, we have no cervids being rehabbed, fawns. And of course, it's unlawful to import live cervids into Oregon. So this is just a real quick case. Why we're concerned. This is Wisconsin. This is an awesome little study. It's 4 counties down here in the southwestern part of the state. Dane, Iowa, Richland, and Sauk counties. Those dots, if you look at those dots, those show wild, it shows a bunch of things on this, but CWD wild positive locations, you know, past positive CWD farms that have been depopulated, all kinds of things in this area. It's a highly infected area. Well, they mapped out via zip codes from those 4 counties for 1 year where all these hunters came from. And these are hunters harvesting an animal. And of course, there's a handful of them here in Oregon, but there's a handful of them in every state. And this doesn't have this. Hawaii had a harvested animal that went back to it. So you know, it just goes to show that how important it is to have good carcass movement regulations. So best management practice, you know, we allow baiting, we allow feeding at different levels with ungulates. Best management practice is not to do this. It's something we have to address. We're going to start working with our taxidermists and meat processors. It's a great surveillance tool, as you can imagine, particularly taxidermists. They're just getting male animals, and a lot of them in a lot of states will collect CWD samples. And I'm going to pass this over to Don now. I think I've already talked too long, rattled on. So.
Okay, thanks, Colin. I'm trying to take over my power grab on the screen here. So. Okay, do we still see the same slide?
We see your Zoom meetings and looks like a backdrop.
Okay, so I must have hit the wrong one here. Let me try this again.
It's all right.
Sharing. Gotta love technology.
There we go.
Now do we see the screen? Okay, so Colin's done most of the talking to this point, but now it's my turn. Let's talk a little bit about the response. As Colin pointed out, it's kind of a 4-pronged response. There we go. Our existing sampling strategy, as he pointed out, is, you know, working with the opportunistic sampling, the check stations. I think I actually started those specifically for CWD about 20 years ago. The roadkill salvage, that's given us annually since that went into place. That's the highest proportion of all the samples that we get annually. And the clinicals, and you know, we're now over 24,000 samples, which is good, but it's maybe not so good. So as Colin's kind of pointed out, really we have to do this within some statistical requirements to ensure what we're doing. And most of the time, you know, we do some fancy statistical math, and it tells us we need to get to sample 3,000 animals if the disease is in an area at a prevalence of 0.1%. And as prevalence in the population increases, the number of samples we need to collect and test drops pretty quickly. So let's turn this around a little bit and let's look at what our new priority area is, Northeast Oregon. If we look at Northeast Oregon and we look at the deer standpoint, we can see within 25 miles, we've got some positives there in Idaho. And this is no news to you guys, but our mule deer population has been struggling for quite some time, and they're on their way down. We're somewhere around, you know, 20,000 deer in Northeast Oregon. Harvest, yeah, we're doing okay. We're holding our own for the last 4 or 5 years. Total harvest bumping a little more than, you know, 2,500 total deer. So the number of deer we need to collect and sample, well, let's talk about elk. Elk are doing, they're just kind of poor, dumb, and happy up there. We've got over nearly 25,000 elk. And we're killing between, 3,000 and 4,000 elk annually just in Northeast Oregon. So most of the hunters up there are pretty happy. So Colin and I have both pointed out that we've statewide collected over 24,000 samples, and we're collecting between 800 and 1,500 samples annually. Well, in the entirety of Northeast Oregon with 4,000 deer and elk harvested annually, we've managed to collect 85 samples from deer and 216 samples from elk. Now, statistics can do a couple of things. One, they can tell us what we need to do, but they can also tell us how well we're doing. And this graph looks a little bit complicated, but it's very, very informative. Each of the colored lines on the graph are representative of how much disease there is in a population, from 0.1% all the way up to 10%. If we take our sample size that we've been collecting in elk and we turn that function around in Northeast Oregon across the whole area, we actually have a 98% probability or 98% chance of actually not finding that disease if it's out there at 1.1%. And this is really a telling piece of information for us as an agency. We've been working hard to collect samples, but when we start looking at the data, we're really not necessarily doing all that great. We don't have a reasonable percent of even detecting the disease with our given sampling effort till it's already at 1%. And by then the horse is already long gone out of the corral. The same thing for deer. Again, we've got nearly a 100% chance of not even knowing the disease is out there with our sample sizes at a low prevalence rate. If we detect it with this sample size, this very small sample size, that disease is really, really, really prevalent out there on that landscape. And that is a really big problem for us. We do have some good news is if it is in that area, we've got lots of movement data from our deer and our elk in the last 10 years or so, 8 years or so. These points are deer. And Interstate 84 is indeed a pretty good barrier from a deer movement standpoint. We do have evidence of mule deer crossing the Snake River and going into Idaho and vice versa. Our colleagues in Idaho pointed out they've got some coming over to visit us. So we know that is indeed a concern. Same thing with elk. There's about 600,000 elk locations on this map right now, and you can see this red line, if you can see it, that's Interstate 84. They're not crossing that interstate very much at all. They do, and we get road truck animals off of I-84 all the time, but essentially it's a barrier to movement. So one of the things that if we do, we're gonna, as Colin's pointed out, we're already stepping up our sampling effort. We're going to focus on the high-risk area in Northeast Oregon, but Idaho was doing that, and it jumped across all over to their border. So that's why it's important that we continue not only with our focal sampling, but we're also going to increase our sampling around the state to try to increase our statistical chances, our scientific chances of actually detecting the disease if we do have it or if and when it gets here. This is going to involve multiple mandatory check stations, and this is a huge thing for us. We've been running the check stations for 20 years, almost 20 years here in the state of Oregon, and we guesstimate, those of us that have done it a long time, that we will maybe see less than 10% of the hunters that drive by us with animals will stop. So these check stations are a huge opportunity for us. As soon as Idaho discovered it. We worked and set up some drop boxes. We've been doing that around the state, but we increased the locations of those. One of the reasons why we're discussing this with you is we're looking for help from our partners, you know, backcountry hunters and anglers, OHA, the bow hunting groups. All of them can be very helpful from our standpoint, both in collaborating with us and providing us samples and helping us get the information out there as far as the importance of why it's so important. And one of the big struggles for us, and it frustrates us at these check stations, is, as Colin's pointed out, this disease is in the animals for a long time before they become clinical. It's always fatal. And what that really translates into is some of the most important animals that we want to get samples from are those older mature animals or the trophy animals. And those are the ones that folks don't want to stop. But realistically, those are the ones that are the most important for us. So it's very critical that we start finding ways, and that's why the taxidermy opportunity is going to be very helpful as well, because as you saw in the pictures, we cut a big hole right in the front of that animal to get what we want. There's some folks that aren't really excited about that. And we're working with all kinds of stuff to try to speed up our processes so that we can get the information back to you guys. And internally, we're already talking about some of the issues within ODFW that we have to try to address. You know, baiting is not prohibited in the state of Oregon for anything except for bears and waterfowl. And upland game birds in some situations. So that's something we have to address. And baiting concentrates animals. If there's infected ones, it's going to concentrate it on the landscape. Feeding is an issue, does the same thing. We ourselves have feed grounds, and we ourselves have feed grounds in Northeast Oregon. So we're having some discussions about how we're going to deal with that. And of, you know, crazy importance is that transporting the carcasses and management of how carcasses and parts of carcasses are disposed of. It's a tough situation to deal with. The good news is for backcountry hunters and anglers, you're leaving a lot of your parts out there on the landscape when you're successful. If you're doing it the way I think you're doing, that's actually a good thing. You're not moving them around on the landscape. That map that Colin showed where we had, what, 6 or 7 individuals that brought positive whitetail deer home from Wisconsin. That's a big issue because most states also notify hunters that their animal's positive. If I brought one of those things into my house, my wife would say, take that out of my house and get rid of it. So it's not just the pieces and parts, but it's also everything after you get it home that we have to start considering, and we have to start educating our people and our constituents and our sportsmen and women just like me. If for some reason we do get a positive, we're going to try to react as quickly as we can. We're going to start with as close as we can get to where the animal actually came from. That's one of the reasons why one of the pieces of information we collect when we collect our samples is the location of that animal. Now at our check stations, and most of our data is collected at the section level, 1 square mile. From a biology of the animal standpoint, that's a pretty narrow or a pretty small piece of real estate. And from there, we'll intensively sample around that area and work our sampling out from the infected zone out into the surveillance and the buffer areas. One of the things that we will try to do as quickly as we possibly can do it is to determine what the prevalence and the distribution of that disease is, because that really is going to have a huge effect on our management. We're currently working on a number of items to try to reduce the points of concentration, the baiting, the feeding. If we do get a positive, as Colin's kind of pointed out, especially with mule deer, prevalence is very high in the mature males or the mature bucks in the mule deer population. I believe it was just last year, there was a very useful publication came out, and it points to the fact that a method for trying to manage the prevalence and distribution that shows a lot of promise is actually increasing the level of harvest on that adult male segment. And we need to sample those that are taken. And so there's a really good chance that if we find it in mule deer, that's where we're going to go. We want to try to manage it. I'm not aware of anybody ever getting rid of it, but there's a lot of different tools we can try to do to try to manage it. And that's where we will go. And we will try as much as we can to use our sportsmen and women to get us down that road, because honestly, it's for you as well. And it's very important that you guys are the ones that are out there that have the opportunity to provide us the most samples that we can get, if we can get the participation. As Colin pointed out, the plan's got a lot of stuff, including the information. We have in our refining some talking points that can be provided, developing those media messages, both in all of the new media that I'm not very good at. And like we're here talking to you guys and we're working with our partners. I think the studies, the human dimension study on these things started easily 15 years ago. We've been working on those a long time. And that actually is an important aspect of all of this, because of the mixed opinions, not only on the disease itself, but there's a lot of mixed opinions on the management as well. It doesn't take a rocket surgeon to figure out that if we go out there and are purposely trying to harvest more males and more adult males in the population, it's not going to be very long and we're going to start seeing some effects on the composition of that population out there on our landscape. And there's a few people that don't like that, just bottom line. And so we get mixed participation in that in some situations, or other states have seen mixed participation in those efforts to try to manage it. How can you help? I mean, can't say enough about give us an opportunity to collect the samples out of every animal that's harvested. It's very, very, very important, especially the mature animals, the older age animals. Those are just, those are the ice cream on our cake here when we're trying to talk sampling from that standpoint. The roadkill stuff, we're going to continue that. We had a bit of a lull there during the COVID years. We relaxed our requirements for testing animals a little bit and only required the males to be brought in. We don't want roadkill to become a loophole for antler collection. So we relaxed it a little bit, but we're back into mandatory for all of that. Get the word out for hunters in Oregon, and importantly, hunters within your ranks and everybody else's ranks that are going to other states. That's really, really important. When I put in the rules for carcass disposal and the live animals and all of that stuff, both of those and the 2 times I went in front of the commission for those actions, they were a little bit bumpy in some cases, especially when we did the live animal thing, because at the time, the research elk from Starkey Experimental Force happened to be in Washington on a study. When the commission adopted those rules, those animals would not have been able to come home. So we had to do some other stuff in there to get that, but we've since fixed that. And then again, all of us as hunters and sportsmen on the landscape, and those of us as managers, we need to do what we can to reduce the spread. You know, carcasses, as Colin said, we're very reluctant to move animals around the landscape. He's pointed out that it's typically done within the recent normal distribution area for those animals. And in some cases where it's not, we have to have long-term historical disease monitoring for some of the other stuff, like we do with sheep. And I don't think Colin mentioned it, I might have missed it. One of the challenges we have with our sampling efforts, and you know, we get asked this question rather frequently, why don't we test them alive? And perhaps Colin questions can address that, but there are a few tests that are becoming available, but logistically and economically, they can be way challenging. So that part of it hasn't realistically been worked out, kind of like the vaccine stuff. Those just aren't options that are out there readily available for us right now. So unless Colin has anything else to add, I think we can close our discussion. I have seen a few items come up in the Q&A chat, I think.
Yeah, there's definitely a few Q&As, and I have a few myself and received a few private messages as well. But Colin, did you have anything else you wanted to add in?
Well, you know, I'll just add two things real quick. Is we really, when we get CWD, we're treating it like that is the first case we've gotten. New York got lucky. They had CWD in kind of a weird scenario with a rehab facility. It was also a taxidermist, and one of the fawns came. It was a fawn, a whitetail fawn that actually tested positive for CWD, and they euthanized those fawns, and they euthanized stuff around the farm, and they didn't find another CWD case for I don't know how many years now, 10 years. 11, 12 years. So they found the needle in the haystack. And that's the way that we're gonna treat this we're going for it. So and that's why we'll sample intensively, at least on a small area until we don't get any samples. And then we'll keep, retesting it. We did that when we got TB, what back in 2001, I think it was, Don, right? In an elk farm. We sampled intensively around that farm for years, and
right,
so
that was Rodeo Creek.
Yeah, and so it's just important to try to make sure that if we do get lucky and we got the first case, then we have a place to focus on. That if we don't find any after that, we need to keep sampling to see if something shows up. But it gives us an idea of where to go to next. So that's one thing he ran. We're going to treat it like the index case is the needle in the haystack. The other thing about sampling with mature animals, in the last year, I harvested a pretty nice blacktail buck out of my yard, and as well, I went to Missouri and harvested a whitetail buck, and on both of those, I was testing the sampling of animals that were large mature males. And so I caped both of them out as far as I could and got it the hide down. With the thought was I'm your average hunter, and I'm going to be able, I want to take a CWD sample from this animal, and I want to mount it. Those were the kind of the premise I was working from. And it took me probably another 15, 20 minutes, kind of working, because when you get around the jaw, it gets hard, but it's doable. And I was able to roll it back completely. And in the Missouri case, I didn't actually take the CWD sample. I rolled it back. I worked on it, got all the way back. And then I picked the skull and the cape up, and I took it to a CWD station, and they removed the lymph nodes. So it's all doable. These are all little challenges we have to work. The picture I showed of just going into and cutting through the hide, right at the pharynx, and we do that in the check stations for folks that aren't going to mount their animal with a hide, and a lot of times the animals are younger that come through that we see. They're 2-year-olds and whatever. Now and then we get big ones in. And when I've been at the check station, I've been able to work with the hunter. Some of these too, roll it back, get a CWD sample without ever touching the hide. And but more often they don't want you to do anything with it. And
yeah, the key to that is, and Colin's right, I've done a number of pretty large bulls and pretty large bucks. If they skin back and they get enough of the neck off of the base of the head, we can just go right down through the tube. those of us that have been doing it long enough, we can go right down through the tube, and oftentimes we can get the obex and or both of the lymph nodes from there. The key is to get the neck off, and that's the big issue. We don't have time at our check stations to skin around the neck if there's a bunch of meat there. We don't have time to do that. But if they get it, if they do a reasonable job of caping that down as far as they possibly can, we can often get the samples, but they don't even stop to let us have a look and try. That's a big frustration for us.
Yeah, and so we hope, and this is where reaching out to folks with backcountry hunters and others is that, if somebody's not comfortable with having that taken right there in the field, that we work with them to get it later. And we get it at the taxidermist, or if they're going to cape the head out, that they bring that part to us eventually, and we can get the lymph nodes and actually test their animal. But we just want everybody to be as proactive and positive as possible with this. And we're doing this for Oregon hunters. We're doing this for Oregon's wildlife.
Well, I really appreciate you guys taking the time and giving the presentation. I mean, there was a lot of details in there, and honestly, there's a number of questions here, a few questions I received privately and a few I had, quite honestly. So before I jump into the actual Q&A, I actually wanted to ask one because I think it sort of touches on some of these other Q&A, because a lot of us hunt out of state, of course. We hunt in Idaho, we hunt in places like that. And Colin, you mentioned that, yes, many of us who, if we're hunting far enough in the backcountry, we're doing what's known as the Gutless Method. So we are not taking the spinal column. We are just taking the meat. We might take, leg bone out, something like that. As far as the head and skull, if we skull cap, what is the recommended way to manage that? Do we then still need to boil that skull before we bring it home, or can we just wash it off with a hose in another state? Or, because I guess that's kind of my question is I think there's a lot of concern from hunters hey, I don't want to bring it back, but I don't know if I can't bring back anything at all other than the meat. And, what do I need to do?
Yeah. So